Imagine the sheer responsibility of releasing a brand new medication out to the public, right? If the tests we use to measure its quality are flawed, well, the consequences can be absolutely disastrous. We're talking about inaccurate dosages that completely fail to treat an illness, or even worse, undetected impurities that could actively harm a patient. The stakes are literally life or death, which is exactly why the pharmaceutical industry just can't rely on blind trust. We need a bulletproof, scientifically rigorous system to prove that our analytical procedures actually do exactly what we say they do.
Welcome to This Explainer. I am super thrilled you're here with us because today we're unlocking the ultimate guide to testing reliability. Okay, let's just dive right into it. We're going to decode the ICH Q2R2 harmonized guideline for the validation of analytical procedures. Think of this document as our global reliability blueprint. It's the absolute gold standard that regulatory bodies everywhere rely on to ensure that, hey, when a lab runs a test on a commercial drug substance or product, those results are absolute fact.
So, here is our roadmap for today. We're going to build a reliable testing method from the ground up. We'll start with the core purpose of validation, then prove specificity, define our ranges and limits, establish accuracy and precision, and finally, we'll wrap up with life cycle and multivariate models. Let's get to it. Section 1. Purpose of procedure validation. Building the foundation. You know, every single test, every formula, and every experiment we're going to discuss today exists to answer just one fundamental question. Is this analytical procedure fit for the intended purpose? Notice how the validation process isn't just some simple one-and-done checklist. It's a continuous, really intelligent loop. You don't just start validating out of thin air. The knowledge you generate during analytical procedure development, which, by the way, is covered in the companion ICH Q14 guideline, feeds directly into your Q2 validation protocol. So your prior knowledge, your system suitability tests, and your development data, they actually form the foundation of your entire validation strategy. It is just a beautifully integrated system. Now, this matrix right here is your primary blueprint. It really grounds us by showing exactly which core tests are required based on what you're actually trying to measure. Like, are you running an identity test? Well, then you absolutely need to prove specificity. Are you testing for impurities or running a quantitative assay? If so, you're going to need to demonstrate your limits, your accuracy, and your precision. The guideline basically takes the guesswork entirely out of the equation for us. Moving on to Section 2, Proving Specificity and Selectivity, the detective story. Think of our analytical procedure as a detective. It has to correctly identify the true analyte while completely ignoring all the other suspects hanging around in the room. So, how exactly do we prove our test isn't easily fooled? Well, we have a few strategic options. The most common way is demonstrating an absence of interference. This means we actively spike our product with known impurities or excipients just to prove our assay results remain totally unaffected. Now, if we don't have those impurities on hand, we might use an orthogonal procedure instead, basically comparing our results against a completely different, highly characterized testing method. And sometimes, we don't even need extra experiments. If we're using a technique like high-resolution mass spectrometry, the technology's inherent ability to resolve isotopes can actually be its own justification. Pretty cool, right? All right, Section 3, Defining Ranges and Limits, Establishing the Boundaries. Now that we know our test is specific, we've got to define its working boundaries. The ICH Q2R2 guideline provides these concrete mathematical parameters, so we know exactly where our reportable range lies, confirmed by proving acceptable response, accuracy, and precision. For instance, if you're running an assay of a product for commercial release, you need to validate a range from 80% to 120% of the declared content. If you're testing content uniformity, that range stretches out from 70% to 130%. So the really crucial takeaway here is that you aren't just proving your test works right at the target dose, you're proving it's completely reliable across the entire expected spectrum of manufacturing variability. But what happens when we're dealing with trace amounts, like, say, a highly toxic impurity? Here we hit a truly critical distinction in analytical science. On the left, we have the detection limit, or DL, and on the right, the quantitation limit, or QL. Think of it like this. The detection limit simply tells you, yep, the impurity is in the room. But the quantitation limit is much stricter. It tells you exactly how much of it is in the room. For basic limit tests, the DL is totally enough. But for quantitative testing, you absolutely must validate the QL. No exceptions. The guideline gives you a really fantastic menu of methodologies for calculating these lower limits. You can use visual evaluation for non-instrumental tests, If your instrument produces baseline noise, you could use a signal-to-noise ratio. That's typically 3:1 for just detecting the analyte, but a much cleaner 10:1 for actually quantifying it. You can also use the standard deviation of your linear response. Look, what matters here isn't memorizing all these dense formulas. It's understanding that whichever method you choose, your quantitation limit should be equal to or below your reporting threshold. And often, it needs to be validated directly by accuracy and precision testing right at that lower limit. Which brings us to Section 4, Establishing Accuracy and Precision, Hitting the Target. To separate two of the most commonly confused metrics in all of analytical science, let's use a classic target practice analogy. Let's start with accuracy. Accuracy is your ability to hit the dead center of the bullseye. It expresses the true closeness of agreement between what you measured and what the actual true value is. We prove we're accurate by comparing our results to highly characterized reference materials, or through rigorous spiking studies. That's where we add a known amount of our drug into a mixture and see if our test can recover exactly 100% of what we put in. Precision, on the other hand, isn't about hitting the center of the bullseye at all. It's about hitting the exact same spot repeatedly, regardless of where that spot is. We prove precision in 3 escalating steps of rigor. First up is repeatability, which means running a minimum of 9 determinations in a short time frame. Second is intermediate precision. This is where we test if our procedure survives the random events of reality, stuff like different analysts, different days, or different equipment. And finally, we have reproducibility, which is checking if entirely different laboratories can get the exact same grouping of hits. Now, what's really interesting about the updated R2 guideline is its holistic approach. While accuracy and precision are traditionally distinct parameters, the guideline actually allows developers to elevate their approach. You can assess their total impact together using a combined performance criterion. By using advanced statistical tools like prediction intervals or tolerance intervals, labs can provide a much more holistic, real-world understanding of the true suitability of their procedure. It's a real game changer. Moving into our final section, Section 5, Life Cycle and Multivariate Models, Elevating the Blueprint. Here, we're basically putting the roof on our reliability blueprint and addressing modern, complex analytical technologies. Modern procedures often use multivariate models, like checking a complex spectrum with many wavelength variables rather than just looking at a single peak. It sounds daunting, for sure, but the guideline demystifies this by breaking the validation into 2 highly logical phases. First is model development, where you use calibration data and internal testing to fine-tune and optimize your algorithm. But the crucial step is phase 2, model validation. You absolutely must test your model using independent challenge samples that were not part of your calibration set. This proves your model actually has predictive, discriminative ability out there in the real world. And remember, a validated analytical procedure is not a static thing. It lives and breathes. As you navigate its life cycle, you're inevitably going to encounter changes. You might swap out a piece of equipment, or maybe you need to transfer the procedure to a brand new manufacturing site. When this happens, science and risk-based principles take over. You might need a partial or even full revalidation, or you might employ co-validation strategies to prove the procedure works seamlessly across multiple laboratories simultaneously. By following the ICH Q2R2 guideline, we aren't just ticking boxes for regulators. We're building a profound foundation of trust, ensuring that every single result is accurate, precise, specific, and scientifically sound. Which leaves us with one final critical thought to ponder. Your analytical procedure might perfectly pass the test today, but is your underlying validation strategy robust enough to withstand the life cycle changes of tomorrow? Thank you for exploring this guideline with me. Keep learning, keep questioning, and keep striving for reliable science.